SwayamPrabha. Digital India. Educated India.
Hello and welcome to our lecture on Introduction to Mechanobiology. So, in the last class we started discussing about focal adhesions, right. So, focal adhesions are those structures which stabilize cell shape and they are those dynamic welds which connect the inside of the cell with the outside of the cell. So, if you draw a cell with a focal adhesion for an adherent cell, you would draw it typically by depicting these adhesions like these dots. Okay. So, these are adhesions of the cell, okay, focal adhesions. Okay. So, what are these? So, if you take an image of cells with focal adhesions in top view, you would get an image. Let us say, if this is a cell, you would have these adhesions spread across. So, what you see is here I have drawn the focal adhesions as some of them are in the periphery of the cell which are relatively bigger in size, and towards the center of the cell I have just drawn them as dots. Okay. So, focal adhesions are composed of more than 100 proteins. So, these are dynamic structures, that is why it enables cells to migrate. So, just like you walk, you put your foot forward and then you put your foot on the ground, okay, and that is equivalent to forming a focal adhesion. So, if your substrate is very slippery or does not provide any friction like glass, then your adhesions are weak, it is difficult to walk on glass. Okay. So, focal adhesions are composed of more than 100 proteins, actually close to 200 or even more. So, the list keeps on growing. You have a broad distribution in sizes. Okay. So, you might have adhesions which are 0.5 to 1 micron square versus 3 to 10 micron square or even larger. Okay. So, this shows you that there are various sizes possible for focal adhesions. So, what are the roles of focal adhesions? Of course, it provides stability to cell shape, okay. It stabilizes the cell, just like it is not possible to stand in water. Why? Because the substrate does not resist. Okay. So, your substrate has to resist the contractile forces that cells exert in order to stabilize cell shape. Okay. And these adhesions are the mechanisms which enable cells to actually sense or see the surroundings. And how does the cell see? Via exerting forces. Okay. So, these adhesions also double up as points through which forces can be exerted by cells in terms of pulling forces or contractile forces, and those were the forces responsible for inducing wrinkles on the substrate when cells are plated on a soft compliant substrate. Okay. So, focal adhesions, they would mediate bidirectional signaling. In other words, you can have outside in, or when the cell is pulling, it is transmitting information about the surroundings from the outside to the inside, or you can have inside out, in which the cell is probably pushing outside. Okay. Both these cases you have focal adhesions. So, it is perhaps not surprising that given the gamut of functions that these focal adhesions perform, that there are so many different proteins present within a focal adhesion. Okay. So, of course, the most important among them is integrins. Okay. So, integrins serve as transmembrane links which integrate the inside to the outside, and that is the reason why they are called integrins. So, they maintain the integrity of ECM cytoskeletal linkages. Okay. Now, integrins are only present in animal cells. So, on the extracellular side, these integrins would bind to various ligands, and on the intracellular side to various proteins. First of all, within the focal adhesion itself, you have multiple proteins which interact with integrins. Okay. And these integrins play important roles in migration, inflammation, homeostasis, so on and so forth. Okay. So, if you look at the structure of integrins, so they are non-covalently associated heterodimers. So, they have two units, alpha and beta, and then hence called a heterodimer, and these units associate in a non-covalent manner depending on the outside signal. Okay. So, these subunits come together when they are active, okay, and this conformational changes in the subunit actually lead to signaling through binding of cytoplasmic proteins. So, you have various combinations of integrins possible, actually 18 alpha and 8 beta subunits have been determined. Okay. So, if you look at the kind of binding you can obtain. So, for example, for binding to collagen you can have various combinations. So, what I have written is beta 1 and then within bracket alpha 1, alpha 2, alpha 10 and alpha 11. This would mean that cells might have alpha 1 beta 1, alpha 2 beta 1, alpha 10 beta 1, alpha 11 beta 1. Similarly, for binding to RGD you can have various combinations, in one in which you can have alpha 5 beta 1, alpha 8 beta 1, or you can have alpha 5 beta 3, alpha 5 beta 5, so on and so forth. Okay. So, integrins, one of the most important function of integrins was found in the case of mediating blood clotting. So, what I have depicted here is platelets or white blood cells which are connected by these blue spring like structures. So, these are fibrinogen which is engaged by two platelets at two opposite ends. Okay. So, at this point of contact between a platelet and the fibrinogen, you actually have an integrin connection. Alpha 2b beta 3 integrin is what binds to fibrinogen. However, when you add this peptide RGD, you see that the clot falls apart. So, this is a clot, blood clot, okay, and when you add RGD it falls apart. Why is that? Because RGD, the affinity for RGD for alpha 2b beta 3 is much higher than the affinity for alpha 2b beta 3 towards fibrinogen. As a consequence of which, because of competitive unbinding, okay, so the clot will fall apart. Okay. So, in a sense blood clotting is mediated by integrins, okay, and integrins are central to so many different functions. Let us look at another focal adhesion protein. This is taline. Okay. So, this is one of the first proteins to get recruited to focal adhesions, okay, and they are actually involved in activating integrins. So, it is perhaps not surprising that taline directly binds to integrins as well as to the actin cytoskeleton. Later on in today we will see how these molecules are located. Okay. So, you will have two different isoforms of taline, taline 1 and taline 2, and these have differential function with regards to cell spreading or cell motility. Now, one interesting aspect of the taline molecule is it binds to another focal adhesion protein, structural protein called vinculin, and what has been shown is taline domains unfold under force and expose binding sites for vinculin, and this recruitment of vinculin binding to taline reinforces focal adhesions. Okay. So, this brings us to the to vinculin. Okay. So, vinculin is a 120 kilodalton protein, again localizing at focal adhesions. Again it interacts with many focal adhesion proteins, actin. Now, if you knock down vinculin, what you see is cells become faster migration. So, suggesting that vinculin actually regulates the adhesiveness of cells to their substrate. Okay. So, if you increase vinculin, that should lead to suppression of cell motility versus cells will become steady and they will form bigger adhesions with their substrate. Okay. So, apart from vinculin and taline, so there is another very critical focal adhesion protein called paxillin. So, again it is localized at focal adhesions, one of the earlier proteins to be recruited to focal adhesion, and what has been observed is when you knock out paxillin in mice, the mice die. So, basically paxillin knockout is embryo lethal, and one of the reasons being that when you knock out paxillin, then morphogenesis or development is stopped. Okay. It is known to interact with other focal adhesion proteins, and interestingly under certain conditions paxillin, in spite of being a focal adhesion protein, it can translocate to the nucleus and regulate other signaling pathways. Okay. So, what you see here, this molecule called FAK is nothing but focal adhesion kinase. Again it has important roles in cell survival, proliferation, adhesion and migration, and what focal adhesion kinase has been shown to do is it regulates cell movement by controlling the turnover of focal adhesions. That is, your adhesions once formed, if they do not break, then the cell cannot propel itself forward. Okay. So, you have to have breakage of adhesions, and that is mediated by focal adhesion kinase. So, in other words, if you knock down the level of focal adhesion kinase in a cell, then the cell will be less motile, even though it has all the other machinery in places. And interestingly, in the case of diseases like cancer, FAC can play both a positive role, that means it leads to potentiation of the disease, or a negative role which will inhibit cancer. So, again it interacts with the signaling protein of paxillin. Okay. So, you have these proteins which kind of bind and interact with each other, one another. Okay. So, these are all, so focal adhesion kinase and paxillin are broadly called integrin signaling molecules. Okay. So, you have integrin signaling molecules, you have structural proteins like vinculin, and you have, so these focal adhesions. Okay. So, when you see an image of a cell, okay, what you will always find is there are, at the end of each of the focal adhesions, they are connected by these big stress fibers. So, these are nothing but stress fibers. Okay. So, it is equivalent to anchoring a rubber band to the substrate via two adhesions. Okay. So, these are your focal adhesions, and this is your string or stress fiber. Okay. So, amount of tension you can put in the string is dictated by how much these adhesions can support. Okay. So, for a cell which exerts a lot of force, if your, so force balance requires adhesions to sustain the tension. Okay. So, if you increase the tension too much, then beyond a certain point the focal adhesions will fall apart. Okay. So, apart from these proteins, okay, you have some cytoskeletal proteins. Okay. So, what you see is these stress fibers, what I have drawn, are structures where actin and myosin come together, and these stress fibers are also put together in, like they stay in place through actin associated proteins, and one of the most important actin associated proteins is this protein called alpha actinin. Okay. Alpha actinin is an actin cross linking protein. So, in cells, apart from alpha actinin, so you have various actin cross linking proteins. Okay. So, you have alpha actinin, I will write alpha A, you can have filamin, you can have formin, okay, and various other proteins. So, you have several different actin cross linking proteins. So, they differ in their sizes, in how they cross link the actin network, so on and so forth. So, as a consequence, their localization within different actin structures varies notably. Okay. So, for actin you have 4 different isoforms of actin. 2 of them, alpha actinin 2 and alpha actinin 3, are specific to muscle cells. The other 2 of them, alpha actinin 1 and alpha actinin 4, are present in all non-muscle cells. So, alpha actinin has been shown to bind to various integrin proteins, including integrins, vinculin, signaling protein called PI3 kinase, and zixin. Okay. Now, what has been observed is alpha actinin 1 and alpha actinin 4 have been found to play opposite roles with regards to motility. So, if you overexpress alpha actinin 1 in fibroblasts, cells become less motile, suggesting that alpha actinin 1 regulates cell motility or negatively regulates cell motility. In contrast, alpha actinin 4 is associated with enhanced motility of breast cancer cells, and it is not just true for breast cancer cells, across multiple different cancer cells, including brain cancer or glioma cells, ovarian cancer cells, pancreatic cancer cells, so on and so forth. Okay. What has also been recently shown is actinin 4 expression in ovarian cancer can be used as a prognostic indicator of independent of clinical stage and histology type. Okay. So, these results suggest that alpha actinin, which is an actin cross-linking protein, is a critical regulator of motility or invasion. Okay. So, this brings us to the question, you have so many different focal adhesion proteins, hundreds of them. Okay. If you were to put together at a single focal adhesion, and we draw the very simple schematic of a focal adhesion like this, just a dot or an ellipse, but it brings to question that they must be spatially organized. It is not that you just put 100 different proteins in a box and the entity will operate as a focal adhesion. So, because they have specific binding to several other proteins, it is important that they are spatially localized. So, how would you address this question of localization at focal adhesion? So, if I were to imagine the focal adhesion as a 3D entity, where are each of these proteins present? Okay. So, to answer this question, if you rely on conventional optical microscopy, you will not be able to answer that question. Okay. So, if you were to stain, if you were to use fluorescence microscopy, so I am just drawing several proteins, all of these, okay, in one channel you might see this, in another channel you might see this, in another channel you might see this, suggesting that they span the entire focal adhesion. Okay. So, this is 3 different channels, you have labeled 3 different proteins, let us say this is vinculin, this is integrin, okay, and this is paxillin. All these proteins will show you this kind of localization. So, it is impossible to know how they are spatially localized within this focal adhesion, and the reason for this is because the resolution, okay, the resolution of normal conventional microscopes is limited by diffraction. Okay. So, what we call is a diffraction limit. Okay. So, how do we define resolution? So, resolution is defined as the shortest distance between 2 points on a specimen that can still be distinguished by the observer or camera as separate entities. So, if you look at these pictures, so this is a representative picture of multiple dots which are positioned close to each other. Okay. And what it appears of a dot will appear as this particular structure or concentric structures. If you replot in 3D, it looks as a point spread function. Okay. It is like a 3D histogram which has a given peak, but it also has a spread as you go far away. So, if these are also called airy discs. Okay. So, if 2 points are relatively spaced apart, so even when you look at these images, there are 2 dense spots representing the actual center point of this dot. So, in this case the dot will be positioned here and here, but the dot will appear like these concentric circles. So, in some cases, okay, if these points are far from each other, you can resolve these points. Okay. This is called the resolution limit. You can see, you can still make out. So, even though the histograms are coalescing or fusing, you can still make out where lies the center point of these dots. However, if you look at this particular point, you cannot distinguish between the 2 points because they have completely converged on each other. Okay. So, the resolution limit of most microscopes, so you call it the diffraction limit. Okay. So, diffraction limit is, okay, you can approximately say lambda by 2, where lambda is the wavelength of light. So, this is not the exact expression, but this is roughly gives you an idea of how much would be the resolution limit. So, most resolution limits of most microscopes are ordered 200 nanometers, which is to say that an object which is less than 200 nanometers would look like 200 nanometers. So, even if you take a particle which is 10 nanometer in size, when you image in the microscope image, it will appear that that particle has a width of order 200 nanometers. Okay. So, which would also mean that if there were 2 particles which were closely spaced, it is impossible to resolve them. So, then it is impossible to, given that proteins have sizes order of nanometers and lesser, you cannot resolve two proteins within a single focal adhesion. You will just get is a blob. Okay. So, to circumvent this problem, scientists have come up with this alternate technique called super resolution microscopy. Okay. Okay. And in this super resolution microscopy, so you can actually detect the 3D position of proteins. And why is that? Because if you look at the localization accuracy, it is within 20 nanometers in xy plane and 10 to 15 nanometers in z direction. Okay. So, this will really help you to identify how these proteins are located along the xyz plane. Okay. So, using this technique, Claire Waterman Storer So, what she did, she took some of the most important focal adhesion proteins in terms of integrin, okay, paxillin, focal adhesion kinase, vinculin, tailin, as well as alpha actinin and actin. Okay. So, what she did was she labeled these proteins. So, you fluorescently label these proteins and then image using super resolution microscopy. So, there can be separate different types of super resolution microscopy. So, some of them are, for example, PALM, STED, etc. Okay. So, these are various techniques wherein you can image super resolution, you can image cells in super resolution. Okay. So, now let us say, okay, so you have a microscope, okay, super resolution. Okay. So, which means that it can detect 10 to 15 nanometers in Z, differences in Z direction. Okay. So, when you take a cell, this is a substrate and this is your cell, and let me draw the integrins. Let us say, so this is your plasma membrane. Okay. So, in side view, the same picture might look something like this. Okay. So, you have a 2D plane and these focal adhesions would look like this. Okay. So, if we have fluorescently labeled integrin, let us say, okay, and we take a box here, a small box here, and we ask the question that in this box, how is the integrin signaling distributed along the Z direction. So, what you can get is, so if I were to draw this as a Z direction, you would get a distribution, okay, and you can find out what is the Z average of integrin. Okay. So, similarly, you can label one by one and you can see. So, for example, compared to integrin, one would expect if you were staining with, let us say, paxillin or or FAC, you have some signaling. Okay. So, this would be FAC or paxillin. So, why is this expected? For the simple reason that so your integrin is a transmembrane protein. Okay. So, integrin being a transmembrane protein from the surface. So, let us say this is my surface. Okay. Of all the focal adhesion proteins, integrin should be the closest one from the surface. Okay. Similarly, FAC or paxillin, both these proteins are known to bind to integrin, and at the same time they do not have transmembrane domain. So, they are still cytoskeletal proteins. Okay. So, you must have them slightly above the integrins. In contrast, okay, actin or alpha actinin should have a broad localization deep inside. Okay. So, this would be actin or alpha actinin. Okay. So, based on this, what Clare-Waterman-Storer showed was you have integrin layer on top of which you had, okay. So, here you had proteins like FAC or paxillin which localized, and this is called the integrin signaling layer. Okay. On top of which this layer was more thicker, you had a force transduction layer. Okay. Now, within the force transduction layer, what she showed was the molecule called tailin is actually localized something like this, okay, with the N terminal protein closer to the integrin signaling layer and the C terminal of the protein being away from it. Okay, it is closer to the actin layer. And on top of this, so here is where vinculin was also there, and on top you have actin and alpha actinin. Okay. So, this shows that there is a gradation or spatial localization of proteins within the focal adhesions. It is not like a blob. Okay. I would like to point you to read the following two papers. This Kanchana Wong et al. This is the Clare-Waterman paper published in Nature 2010, and the other paper Patla et al. Nature Cell Biology 2010. Thank you for your attention.